questions for everyone today, just because I felt like we needed it. Last day. So help yourself. I don't want them anymore.
So,
not quite, but close.
I like it. Fine.
So, for our project, we did measure stress response to acidification, pH change in Pacific oyster. Just some background, you guys, most of you know, or all of you know what acidification is, but H2O, or CO2, gets into the water, it reacts with H2O, forms carbonic acid, and changes the pH balance. Pacific oysters are native to Japan, but we grow them here and all around the world for aquaculture purposes. They're pretty temperature tolerant for oyster species. They are, yeah, like most bivalves, they don't have extreme pH changes.
The objective of our study was to look at the physiological responses of Pacific oysters in acidic conditions. So we did this by measuring gene expression and protein expression. And Len looked at metallothioneins, which is it's a general stress response. And I looked at glycine-2, which is used for calcification and shell formation. Morgan looked at HSP70 gene expression, while Dan looked at HSP70 protein expression.
So for our experimental design, we wanted to study the effects of ocean acidification on oysters. And the way that we planned to do it was that we took two 55-gallon barrels. We filled them each about halfway to 3/4 with seawater. And then in one of them, we bubbled in carbon dioxide, and we measured the pH. It is relatively constant around a pH of 7.24 for the control and 5.24 for the one with the carbon dioxide in it. And then the next thing that we did was we set up those tanks, and then we got oysters. We got 18 diploid oysters from Big Beef Creek, Washington, and 15 triploids from Taylor Shellfish Farms. And then we split them up accordingly, where we put 12 diploids and 10 triploids into the control group, and then we put 6 diploids and 5 triploids into the carbonated seawater. And then after 24 hours in the carbonated seawater conditions, all the oysters pretty much died, so we sampled it. And then in the control one, after 120 hours or 5 days, we split them up. So we had 6 diploids and 5 triploids in the dry condition, where we just took them out and put them on the shelf. The other ones remained in the wet control. And then after 24 hours, we sampled them, and when we sampled them, we got 3 gill tissues and 1 mantle per individual.
There are many methods used in analyzing the data that we received, or the tissue we received. A lot of us used qPCR for gene expression, so measured the amplification of the gene we were looking at. Dan, the protein guy, did a lot of STS-PH Western blot to try and figure out how much of the proteins they had, the concentration, and the nucleotide length.
So for mine, this is what it looked like. You can see over here, banding down at, this is 6 kilodaltons, and so it's kind of around at the lower, or smaller size proteins. So they were all diploid, and then there were 2 from the low pH, 2, and then 3 from the high pH. 2 that were left out to dry, and then 1 that stayed wet, or submerged in the water. And so over here you can kind of see, this is what it would look like if it bonded with HSP70, but we had banding down here, or I did, so means that the antibody was going to something else. So it didn't really quite work out the way I planned, but it could be that what happened was it binded with ubiquitin, which, as its name implies, is found in most cells. It's basically just a marker to proteins that they need to be degraded or repaired, and so the chaperones can identify that and come in and repair them. So it wouldn't make sense then why you saw them in almost all the samples.
I did HSP70 gene expression, and this is a graph of the normalized gene expression for diploid wets, which is 2NW, triploid wets, 3NW, 2N dry, which is diploid dry, and then 3ND, which is triploid dry. And so I used the wet environments as a sort of control and the dry environments as the treatment, mostly because when they close their shell, it creates a more acidic environment, so I used that as my stressor. And then I used elongation factor to level out the HSP70 amounts in each oyster and normalize the data. By looking at the data, you can see that it's kind of skewed. It's not exactly what I was expecting, as we kind of talked about in class. So if you look, triploids are supposedly more resistant because they're sterile, and so that energy that would go towards reproduction can be focused towards the immune system. That kind of confused me because I would have thought that the triploid oysters would have had lower amounts of HSP70 in both environments, but they didn't. And the diploid oysters at least had an increase in HSP70, which is what you would expect. So then I looked at taking all of the wet oysters versus all of the dry oysters. So you have triploid and diploid in each category. And you can kind of see that HSP70 decreases when it gets drier, which again is not at all what I thought would happen. And to kind of as an overarching thing, the results weren't significant. So probably if I was going to do it again, I would use a lot more samples and a little bit different setup and test everything all over again. But there were some hypotheses that we came up with in class for maybe why HSP70 would be lower in a dry environment. Some people mentioned that perhaps the dry environment was closer to a control or closer to their norm. And if that were the case, it could be that HSP70 was decreased because it wasn't stressful enough. And in oysters, they have something called a heat shock cognate, which doesn't have the same primer as HSP70 does, so you're not measuring the cognate expression in dry. Another, kind of the polar opposite hypothesis would be that it was so stressed that it just shut down and was pretty much on its way to dying. So, like I said, more testing needs to be done to see what exactly happened, but it's kind of a good start for future experiments.
Okay, so I looked at how the acidic conditions of dry versus wet affected calcification. And to do this, I measured the gene expression of digoxin-2, which is used in calcification and shell formation. As we can see, I used the same two groups that Morgan did, with the wet as the control and dry as the treatment group. And since they build up the CO2, it stresses them. And as we can see, the expression in the dry group was less than that in the wet, which was as expected, because studies show that over time, when a shell or an oyster is in acidic conditions, it tries to maintain an internal pH by slowing the metabolic rate and also the rate of calcification. And so it slows the expression of this gene.
For mine, my study had two hypotheses. It's increasing acidification should upregulate metallothionein gene expression, and also there should be a difference between gene expression for diploids and triploids. So here we see one analysis of the control pH diploids and triploids, and then with a t-test on the bottom right, more to come on that. And then the next one is just the same analysis on the other experimental group, the low pH. And then this one is comparing the diploids in each experimental condition as well as the triploids in each experimental condition. So the results here, I just pulled out all my t-test values for each test that I did. And as you can see, the t calculator right here, in each case is lower than the critical value. And the p-value, we generally wanted around 0.05. And what that means is if it's below it, it would indicate that the results are significant and that there is a statistical difference between the groups. However, in all the cases, it's that's not the case. So there's no statistical difference between diploids and triploids in experimental conditions, and as well as between acidic groups. So here are my hypotheses again, and the first one was unsupported, as well as the second one. And then reflecting on it, something to note was that the individuals in the pH of 5.24 died within 24 hours. So a possible reason for the results that we got was that maybe the oysters died in 6 hours, and then when we sampled at 24, a lot of the mRNA from metallothioneins had degraded. So then the results that we got might be misinterpreted. So the gene expression could have been a lot higher if we had sampled at maybe 6 hours versus the 24 hours. And then nonetheless, metallothionein gene was expressed, so maybe the metallothionein gene expression was a response to the acidified conditions. So then what we would do in future studies is maybe change the time span, either have a longer time to allow the oysters to acclimatize, or maybe do shorter time spans to get a more accurate depiction of the tissues. And then also to have a different pH, maybe have a control pH that's closer to the ocean water, which is slightly basic, then have a little bit more moderate acidities. Thank you.
Are there any questions about